Welcome to session 3 on hematologic malignancies. My name is Tim Graubert. I'll be moderating this session. We have 3 talks, 2 by rising stars in this field. But before we get to that, and I'll introduce them, I'll give the first talk. Last year at the colloquium, there were 3 fantastic talks on lymphoid biology malignancy by Lou Stout, Andy Lane, Bob Copeland. And so this year, there's definitely a myeloid bias to the talks, and that reflects my own area of interest. So, but I think it'll be an interesting session. So I'm going to give the first talk, as I said. Title is here on the molecular profile of acute myeloid leukemia, or AML. No relevant disclosures to mention here. Okay, so I cannot give a talk on genetics of acute leukemia without first acknowledging the really pioneering contributions of Janet Rowley. Well-deserved. This audience obviously recognizes her incredible contributions to the field. It was a real tragedy that we lost her last year, but you see her here receiving the Presidential Medal of Freedom from Barack Obama. She had an amazing career, unconventional in many ways, but really, in my view, established the field of cancer genetics, and as much as anybody else, I think, taught us that cancer is fundamentally a genetic disease. And but I think it's fair to say it was not an easy path for her. There was enormous skepticism. This paper here, published in 1973, I think it says, was the first description of a non-random, a recurrent chromosomal translocation in human leukemia, the 821 translocation associated with a particular subtype of AML. And I understand this paper was rejected everywhere Janet sent it. It finally wound up in a fairly obscure journal in France, where I think it remains the most highly cited publication in that journal ever. And so there was just, the dogma at the time was that chromosomal abnormalities could not be causal. These were consequence of the cancer transformation process, and that this was all noise. So really almost single-handedly, Dr. Rowley overcame that skepticism, identifying this translocation, subsequently, shortly thereafter, showing that the Philadelphia chromosome and CML was a consequence of the 922 translocation, and then in a series of papers over a long career, really defining all of the major chromosomal abnormalities that we now recognize as important in leukemia, including 2 other subtypes of acute myeloid leukemia shown here, the 1517 and version 16. You can see that she eventually won the field over as the trajectory of journals, moved up in the right direction here over her career. So we've obviously learned a lot about these particular translocations in the following decades, and I'm not going to go into any of that in any detail, just make some general observations. The striking thing about these 3 in particular is that they're all balanced reciprocal translocations involving lineage-specific transcription factors important for hematopoiesis. They're acquired early in the course of the disease, and are completely mutually exclusive. You see in this heat map, little piece of a figure I'll show you in a minute from the TCGA paper on AML published last year. But you can see these 3 translocations, the fusion proteins or transcripts formed by them, the first 3 lines here, patient as a column. If you get one, you never get another one. So they're completely mutually exclusive, but we know from many lines of evidence they're not sufficient to cause the disease they initiated, but require additional cooperating mutations. And now, over the last few years of high throughput next generation sequencing, we obviously know a lot about the patterns of mutations that tend to co-occur or not co-occur with these translocations. One initial observation that kind of echoes what you heard this morning from Dr. Rosenberg. This is kind of the same sort of data, the burden of mutations and mutation frequency per case across different tumor types. It's shown a little differently in this figure from a review that Dr. Vogelstein wrote. The take-home point for me, the point I'm showing you this, is that MDS, which we'll hear about in the second talk, and AML have far fewer mutations in the coding region per case compared to, as you heard, melanoma or lung cancer or most solid tumors in general. In fact, really the heme malignancies that have been sequenced are all on this end of the spectrum, closest to pediatric cancers, which have the lowest frequency of mutations. So maybe this is just because we haven't sequenced enough tumors. We don't. This is just an artifact, and we haven't discovered them all. That's possible, but I don't think so. This is a paper again from Getty Getz and colleagues published last year that shows you the number of cases that you need to sequence, so patients here, as a function of the mutation frequency in those tumors. And so AML, again, over here on the left, on the low end of mutation frequency, you need fewer cases to get over these different hurdles, finding all the mutations that are occurring in 10% or more of cases, 5%, and so forth. So I think we probably have almost certainly identified all the very highly recurrent mutations in AML, even with only 200 cases fully characterized and reported, which is pretty much where we stand today. But it's likely that we haven't found all of them, but probably all the highly frequently mutated ones. And so what have we learned from even just a few hundred cases of fully sequenced tumors? This is a disease of incredible genetic heterogeneity, which is a recurring theme in most cancers. This is the rest of that heat map. I showed you just the sliver at the top. I know you can't read the names of the genes here, but genes on rows, again, patients in columns, and cells here showing patients with mutations. And the point I'm making is simply that, of course, AML is not one disease, just like lung cancer, breast cancer, stomach cancer is not one disease, but there are many genetically defined subtypes within AML. Again, even with only 200 cases, which is what was included in the study. So with 200 cases, there were 260 genes that were mutated in 2 or more patients. So an enormous amount of heterogeneity between patients. And even after correcting for statistical significance, the size of the gene and so forth, 23 genes, relatively high number of genes, so-called significantly mutated in this disease, and that's what's shown here. And this again is a recurring theme in cancer biology where we have a handful of genes highly recurrently mutated. The first 2 here are FLT3 and NPM, which we've known about for some time in AML, and then the dreaded long tail here of genes headed off in this direction that are many of which are going to be important biologically, but are more and more difficult to study as they're less common. So some general implications, I think, from this level, looking at the genome data in AML. It's going to take a lot of work, I think, to show which of these genes have prognostic significance in this disease. I think it took close to 2 decades to really parse out just those first 2 genes, many thousands of cases, just to really understand the interaction between FLT3 and nucleophosmin mutations. So we're going to hear more about this. Dr. Levine has made contributions here, but it's going to take a lot of time in many cases to really grind through all this data and figure out which genes have prognostic significance, which are potential therapeutic targets, and how are we going to test for this routinely in the clinic with such a large number of recurrently mutated genes. So I'm really not going to dwell on any of these points, but these are all challenges for the future. Other implications, again, from this genetic heterogeneity, I think it will inform better, more faithful genetically engineered mouse models. I think, again, Dr. Levine will echo this theme. In my mind, it makes most sense to design your genetically engineered models in a way to recapitulate the pattern of mutations that co-occur or don't co-occur from patients. And obviously, this has provided new insights into the biology of this disease. So first thing I want to dwell on for a little bit here is one particular pathway involved in RNA splicing. That's on the bottom line of this figure here. You can see that various genes encoding factors that are important for RNA splicing are recurrently mutated in AML. One in particular is this factor U2AF1. You probably can't see it. It's in the middle here, mutated in about 10% of patients with AML. So I'm talking about this because I think for most people this was not a biological pathway that was on the radar screen when we thought about cancer biology. But these genes are highly mutated, mutated at a very high frequency. Most patients with MDS, the disease we're going to hear about, have a mutation in an RNA splicing factor. They're mutated across the spectrum of heme malignancies and now in solid tumors as well, in lung cancer, breast cancer, pancreatic cancer, melanoma. So we need to think about these and what the biology is telling us. So a couple other general comments about these RNA splicing factors. This cartoon here just reminds you that the spliceosome is a molecular machine that comes in and defines introns, shown here, and excises them, which gives us a lot of combinatorial diversity in our transcriptome from one genome. And here again in the simplified cartoon, spliceosome, complicated machine, 200 proteins or so, just a few are shown here. But the ones in red are ones that are recurrently mutated. Again, they're early, acquired early in the pathogenesis of the disease. They're almost perfectly mutually exclusive, like the fusion genes. You acquire one, you rarely, if ever, acquire a second one. And as you can see in this, even in this cartoon as well, they are not randomly distributed across the spliceosome. They're highly enriched here in the U2 SNRP, the portion of the spliceosome that defines the branch point and the 3' end of introns, which form a lariat and subsequent excision of the intron. So what do these mutations do? How do they contribute to cancer? Very much a work in progress. Obviously, the working hypothesis has been, since, again, the normal role of this complex is to mediate alternative splicing, gives diversity here, that these mutations must contribute to cancer in some way by perturbing that, to changing the pattern of alternative splicing, and somehow that rewires cells to give them a growth advantage. That's the hypothesis. So again, I'm going to show you a little bit of data. We study this factor U2AF1. It's part of this complex. It recognizes that those last two nucleotides, the AG at the end of introns, which is present in virtually all of our introns. This is a cartoon of that protein. The interesting thing about these mutations is they're all missense substitutions, almost all of them in just two codons, serine 34, glutamine 157, that are conserved down to yeast and are in two predicted zinc finger regions of the protein. More than 80% of the mutations are this particular allele, the S34F substitution, and that's what we've focused on. I'm going to zip through a little bit of data that is largely in a paper that just came online last week, but we wanted to really study in a genetically defined system what are the consequences on splicing when you overexpress this mutant allele compared to the wild type. So to do that, we took normal human hematopoietic cells and we introduced either the wild type or the mutant, this most common allele, the S34 version of U2AF1, or an empty vector, into these cells, and then did RNA-seq, identified alternatively spliced transcripts, and then went through a variety of stages of validation after that. And the bottom line is we see an enormous amount of alternative splicing that's dependent on this mutant protein. In something like 6% of all transcripts that are expressed in these cells were alternatively spliced as a function of expression of the mutant allele. This is one class of these, the junctions that we were measuring in this study. Every dot here is a different spliced junction. And you can see that some of them are increased in abundance when you overexpress the mutant, some of them are decreased. And all classes that we looked at were similarly distributed in this way. So it's not just increased, again, it goes in both directions. These are real, so these are findings from RNA-seq. We took a large number of these and validated them just by RT-PCR in different samples. Here are a couple of examples. So here's an event in the DEC oncogene where there's a cassette exon that can be included or skipped, forming these two isoforms. Here's another event where there's a cryptic splice site that can be used or not used. And so from the RNA-seq data, we found increased abundance of these two alternative transcripts. And you can see from RT-PCR in different samples, we see the same thing. And not only are these present in these model systems, they're present in actual patient samples. So that's what's shown on the bottom. These are cells from patients with MDS who either do have the S34F mutation, those are the filled bars there, or lack all mutations in splicing factors. And you can see there's a nice genotype dependent segregation, again, with increased alternative splicing in samples that have this particular mutation. So again, not only in these model systems, but these are present in fully transformed primary cells from patients. One interesting feature about these alternatively spliced events, it's not random. There's some sequence context that seems to matter. This has been seen now by several groups. So it's hard to see here, I know, but I'll walk you through it quickly. These are logos plots. So if you focus here on the right, these are junctions from the RNA-seq data that were not changed, mutant compared to wild type, so controls. And you can see that for the most part, you see, first of all, the height of a logos plot indicates the frequency of that nucleotide. So in all cases, it's AG, again, at the end of an intron. The nucleotide immediately preceding that is usually a C in junctions that are not changed. But in junctions where the mutant protein likes to skip, either a skipped exon or a cryptic site that it skips, there's a change in the abundance, now a T instead of a C in this position, which we don't see with the wild type, suggesting that the mutant protein, perhaps the affinity of this protein binding to these sequences is different and may have something to do with which junctions it chooses to skip or not. We were able to provide some evidence that supports that. These are studies in collaboration with Clara Kielkopf in Rochester. So Clara designed a number of oligos that corresponded to the sequences in the introns of some of these skipped events that we saw. So here again is this DEC oncogene where this is the cassette exon that can be retained or skipped. So here are the sequences of the AG and the preceding nucleotide and the flanking nucleotides, again, that the mutant likes to skip. And then here's the normal sequence of the downstream junction that it prefers to splice into. And so she made recombinant proteins, the U2AF protein, wild type or mutant, with cofactors, and then took this complex and incubated it with these oligonucleotides and did binding assays and showed results that are really consistent with the prediction I just made a minute ago. The junction that the mutant enzyme likes to skip has lower affinity compared to the wild type protein, whereas the junction that it skips into is equivalent to the wild type, has no preference. And then when we mutated those sites to these oligos shown on the bottom, it caused the reciprocal effect, and we've seen this for a number of the junctions. So the sequence context seems to affect binding affinity of the mutant compared to the wild type factor, and that contributes to alternative splicing in these cells. So much more to come, I think, on the mechanism, how these splicing factors are working. Many people are working in this field. You may hear something from Dr. Ebert in a minute on a different splicing factor. Next, we wanted to ask, does this have any consequences? Find these mutations change splicing, but does it have anything to do with hematologic disease? Does it change hematopoiesis? So here, we've used a mouse model that my collaborator, Matt Walter, has generated, where a single copy of the human, either wild type or mutant protein, U2AF1, is knocked into a locus that's under the control of doxycycline, so you can turn on or off at any desired level by titrating the dox, this protein in these two different strains of mice. We've characterized these mice quite a bit. I'm just going to show you two observations. One is here. So this is a short-term exposure, just 5 days of exposure to dox in these mice, turning on the mutant protein or the wild-type protein, and we see changes in hematopoiesis. Increased numbers of progenitor cells, that's shown two ways here, one by flow cytometry, and I'm showing you the mutant cells here compared to the wild-type or other controls, or progenitor cells grown in methylcellulose. So just 5 days of turning on this protein changes hematopoietic differentiation. Then the other thing I want to show you, in these mice, as you predict, splicing is also altered. We've looked at this comprehensively. I'm just showing you really one example here that illustrates generally what we're seeing. This is the FMR1 gene, not a gene that we think has anything to do with the biology of MDS or AML, but this is a well-credentialed alternative splicing event that's been studied extensively. Here there are two cryptic sites, so you can have 3 different transcripts, wild type with these 2 different alternative forms. And in every system we've looked at, when there's the U2AF1 mutation, we see aberrant splicing of this transcript, so increase again of these alternative forms, either in patient samples with MDS or AML or these transgenic mice. you see again increase of these alternative forms in the mutant compared to the wild type. Okay, so now a bit of a left turn. So given the fact that this mutation changes splicing, affects hematopoiesis, could we begin to think about perturbing the system and potentially reversing some of these phenotypes? So there are a number of compounds that have been studied for several years that perturb splicing. The parental compounds are actually naturally occurring. These are bacterial products that have been derivatized in a number of ways. One by Eisai, now H3 Biomedicine. This is a drug that actually went into the clinic, E7107. We focus mainly on this class of compounds called Sudemycins that a medicinal chemist at St. Jude, now at SRI, synthesized. And these molecules bind the splicing complex, change splicing, and are useful for preclinical studies. So we're returning to this mouse model I just showed you, where in this system I showed you just this 5-day pulse of expression of the mutant protein changed splicing and hematopoietic progenitor numbers. So the question here is, what does this drug now do? Here again is the data that I showed you, different cohorts of mice, but the same observation. 5 days exposure increases these progenitors. And down here, this is a different control cohort without drug, but now you see with the drug, we see normalization of these progenitors. This is 5 days of exposure of Sudemycin and 5 days exposure to the mutant protein using both of these assays. And the splicing abnormality that I just showed you is also reverted in these mice that we treat with this drug. So again, early days, but I just wanted to share with you again an interesting pathway that really I think we wouldn't have been focusing on were it not for these unbiased genomic approaches to studying AML. Okay, so I focused on the genetic heterogeneity between patients with AML. I want to now turn to the heterogeneity that we see within individual patients with AML. And again, this is a general theme that many of us have seen in different types of cancer. So we initially approached this looking at patients with MDS who then progress to AML. This is an event that happens in about a third of patients with MDS. And we looked by whole genome sequencing at these 2 time points, and we're able to identify all the somatic mutations present at these 2 stages, and with a large number of genetic markers could group these together and identify a population of cells where these mutations are traveling together. This first clone we call the founding clone. So in all cases of these AMLs derived from a pre-existing MDS, the founding clone was still present and detectable when the patients progress. But again, in all cases, new subclones emerge, which appear to be derived from the founding clones. So one cell here acquires additional mutations, in this case 112, and becomes the dominant clone at AML. There are a number of assumptions here, and I wanted to spend a couple of minutes kind of drilling down on this and showing you some new data that support this model. So this is actually the kind of data that lies behind that plot I just showed you, which is plots are kind of intuitive, but it's hard to understand exactly where those come from. They essentially come from data like this. So here again are all the somatic mutations detected and then validated in these patients. This is one patient, so every dot here is a somatic mutation. And I'm showing you the allele frequency, the proportion of reads that detect a particular mutation when the patient had MDS, and then in this axis when they progressed to AML. So assumption number 1, if we can cluster these mutations, identify mutations that have, quote, similar allele frequencies, that implies to us that these are actually traveling together in a clonal population of cells. We use a variety of tools to do this clustering. Cyclone is one of our particular favorites at the moment. So Cyclone would say that this is the founding clone present when the patient had MDS. When the patient progressed, the founding clone is still present, but now there's a subclone. And again, assumption 2, all of these cells that bear these mutations also still carry the founding clone mutations, are all carried forward. So that's probably clear as mud here, but let me go on and explain how we follow this up. This summarizes what I just told you here. The data largely support this kind of linear evolution model where normal cells acquire mutations. I'm going to come back to this point in a bit. But when this first founding clone is established with disease, it carries forward, again, all of these pre-existing mutations, and then subclones derived from the founding clone, again, and have new mutations as well. So again, these two assumptions that I just want to address for a minute here. So we did this by looking at single cells in these patients. So we selected a number of patients where we had already sequenced the tumors at MDS and at AML. We knew what the somatic mutations are. And so now we, again, want to look at single cells and see if we can support the model that I just showed you a second ago. So we identified these, isolated these single cells, amplified the genome, and then did hybridization, capture, and sequencing again under the lamppost here. We're not trying to discover new mutations. We're looking where we already know that there are mutations. This work, again, was published recently, so I'm going through kind of quick. So this is the data from the new hybridization capture, but not from the sorted samples. This is just going back again to the unsorted MDS and AML samples. I'm just showing you that this, the strategy works. There were several hundred, more than 300 mutations that we said were traveling together in this initial founding clone, present at both MDS and AML, but absent in the skin, which we use to subtract germline polymorphisms. And then another 369 mutations that we were saying are in the subclone, only at AML, absent in MDS. And you can see here we had paired blood and bone marrow samples that were concordant. So that's the unsorted bulk material at the top here. At the bottom, you have a dozen single cells. And so first of all, just to point out a couple problems, but, which I'm now going to dismiss. One is that in light gray, there are a lot of cells that are empty, simply didn't give high quality sequence. And the second dark gray, we could sequence the area, but we only saw the wild type allele. We didn't see the mutant, so this is due to allele dropout. This is an artifact. This is a problem of whole genome amplification where there's dropout of sites entirely or one allele or the other, which is this huge problem if you're, although the techniques are getting better, but if you're using this to try to identify mutations, there's a lot of missing data. When we know the mutations we're looking for, we're going to essentially ignore those holes, and really only the colored cells here are informative. And it shows that all 12 of these clones that we obtained from the AML patient were part of the subclone. These mutations travel together and derive from the founding clone again, because you never see these subclone mutations in single cells that don't have the pre-existing founding clone mutations. That's a simple case. This is a more complex one with 5 subclones. Here's the unsorted material. Here's a dozen sorted cells underneath, and you can see different stages of this clonal hierarchy that are represented by the different single cells. Again, largely supporting this idea that the clusters come as a block and carry forward with them all of the pre-existing upstream clusters of mutations. Okay, so I think going forward, this is it will be interesting. Many people are doing this, of course, thinking about these models of clonal evolution in the context of the therapies that we're giving our patients, and what is the impact of therapies, and which clone should we target and which one should we not target. This is an early study led by Tim Lay, where we looked at again AML patients given standard cytotoxic chemotherapy. Here, all these patients went into remission, but all of them relapsed subsequently, and we saw a number of different patterns, but the general themes are illustrated here. When you sequence the genomes at diagnosis, we would often see a number of clones, the founding clone here in gray, and then subclones. Some of these subclones were extinguished and never came back, but in all the patients that relapsed, the founding clone was always detectable again, and the dominant clone that emerged was always derived again from the founding clone, really suggesting that the problem with relapse in AML is simply our failure to eliminate the founding clone. So in the last couple of minutes here, what I'm going to focus on is really these earlier events, and what I want to do is set the stage for a theme that I think is going to resonate to the other two talks in this session. So what's happening here before a founding clone is established. So there were a couple of papers that were published several years ago in Nature Genetics that really, I think, started this story. So this is, both these studies used data from SNP arrays done on peripheral blood samples from patients, either patients with cancer or without cancer. For the purpose of what I'm showing here, it doesn't really matter. But they identified somatic mutations in these ostensibly normal blood samples. These are large structural variants that had to be 2 megabases or larger. either copy number abnormalities, gains and losses, or LOH events. And the basic observation shown here in this histogram, which is similar in both these papers, that these structural variants were detectable in normal peripheral blood samples from these individuals, and that the frequency in which you can detect them rose with age. So it's interesting. So this is mosaicism in the blood of individuals who don't have blood cancers. So I think this story, the follow-up to the story is a paper that one of our distinguished panelists published shortly thereafter in collaboration with Lambert Busk in Canada. This is a study of older women with skewed hematopoiesis, clonal hematopoiesis, defined by X inactivation. So Dr. Levine and colleagues studied sequenced the exomes of a number of these patients, found TET2 mutations, and then extended in a larger cohort of patients and found mutations in this gene at a very high frequency, 5% or more, in these women who were, again, normal. They had skewed hematopoiesis. You can see here their blood counts are perfectly normal, indistinguishable whether they had TET2 wild type or mutations. Again, building this idea that at a relatively high frequency, there are people walking around with mutations now in bona fide leukemia genes that are emerging to the level of detection. And by definition, they're clonal in the blood of these people. So to take this one step further, but not all the way, because we're going to hear more on this theme in a minute, my former colleagues at WashU recently published a paper, this is online now in Nature Medicine, where they examined TCGA data, again looking at the peripheral blood samples that were used for most of the TCGA samples, sequencing studies as the normal, a surrogate for the germline. But looking at that normal peripheral blood sequencing data, they again found clonal mutations at a very high frequency on these patients, on the order of 6 to 7% now, in patients over the age of 70. And the amazing thing is that 83% of the mutations that were clonal in the blood of these individuals were in known leukemia genes. And that's shown in this rose plot here, DNMT3, TET2, et cetera, going around here. And again, increasing frequency with age in these individuals. So the model that emerges from this is that all of us, we know this now from direct evidence, are acquiring mutations in our hematopoietic stem cells, all of us, and they increase with age. In a surprisingly high percentage, maybe 1 in 20 individuals, mutations are acquired that are sufficient to establish clonal hematopoiesis. Now, most of these people don't go on and get leukemia, right? This is still at least an order of magnitude higher than the frequency of leukemia in these in these individuals. But it suggests that this is a model for progression, and I think raises maybe a general theme here. We've known about this progression for some time in myeloma, where there's a precursor lesion called MGUS, monoclonal gammopathy of undetermined significance, where people walking around normal have clonal populations of plasma cells in their blood secreting monoclonal paraprotein. And we know that there's about a 1% per year risk of these patients progressing from MGUS to myeloma. More recently described as an analogous process in CLL, so just described about 3 or 4 years ago, where there's frequently a monoclonal B-cell lymphocytosis phase that again appears to be a precursor lesion for CLL. And then now we have this evidence of clonal hematopoiesis leading to myeloid leukemias, MDS and AML. Lots of questions that we don't know yet. Is this a required progression? Do all patients with MDS and AML have this precursor lesion? What is the risk of that evolution? Should we care? Should we do anything about it? Or should we be surveying people? Should we even be thinking about intervening? I think these are all questions that we'll be thinking about going forward. Let me just summarize what I just showed you here. This shows, again, normal stem cells accumulating these background mutations, which we showed previously in a paper that, again, accumulate from cord blood to individuals in their 60s and 70s. But on the order of 5, 6, 7% of people, mutations, particularly in these epigenetic regulators, are acquired, resulting in clonal hematopoiesis, not frank disease, but then perhaps, again, if this is really a progression, a subset of these patients acquire true drivers that are sufficient to establish the founding clone in this disease, And interestingly, these tend to be different. These are mutations that tend to be present in the founding clone, not the genes that we tend to see here early in clonal hematopoiesis. And then in progression from MDS to AML or AML at diagnosis to subclones, additional cooperating drivers cause expansion and the clones to move forward. So just to summarize what I've told you today, AML is a disease of genetic heterogeneity, which we all know now, cancer in general, between patients and also within patients at the level of intratumoral clonal heterogeneity. Talked a little bit about these spliceosome mutations, which I just want to make sure it's on everybody's radar screen, an interesting area of new biology that we still need to do a lot of work on to understand mechanism and potentially think about them as therapeutic targets. And then this interesting observation of somatic mosaicism in the blood system that seems to be quite common and increases with age, may or may not be a precursor to the development of leukemia. I'd conclude from this, we've learned a ton from sequencing just a few AML genomes. Lots of work still to do to refine, to use this information to refine prognosis, pick therapeutic targets, and work out the biology. I kind of alluded to this, and I think we'll hear more tomorrow about routine genetic profiling, but the standard of care today in this disease is to look at 3 genes, maybe 4 genes. But we know now that there are many more genes that are recurrently mutated and important in the biology of this disease. We need to figure out how to do this, bring that information, to our patients in a more routine way. And again, this clonal heterogeneity issue, I think, is going to be very important in terms of picking targets that are in founding clones versus subclones, monitoring patients, and thinking about this issue of clonal hematopoiesis before and after treatment of leukemia. So I'm going to stop there, thank my former colleagues at WashU, where most of the work was done that I talked about today, especially Tim Lay, Rick Wilson, Matt Walters, my collaborator on MDS studies. And my new colleagues here, having been here now at MGH for a year, I'm delighted to have this great group of leukemia investigators and collaborators in pathology. And I wanted to point out here, I am now connected to the Bertino-Chabner lineage because I was lucky enough to recruit Joe's daughter Amy as my first hire in my lab, and so I think I'm very, very fortunate to be in that company. All right, let me leave it there and see if there are any questions. Thanks very much. Probably have time for a couple questions.
I guess the big question is, are these patients who have these pre-existing mutations, are they in a higher percentage of the, do we have?
Yeah, Joe asked the question, so the patients with clonal hematopoiesis in the blood, is it really kind of, I alluded to, is this a precursor, or are they at risk, in fact, to progress to MDS or AML? What is that risk? I'm going to give a brief answer because, again, this is a theme that we're going to hear more about in the next 2 talks. I think we don't know. I know there are studies that are being done now with really incredibly valuable retrospective repositories of samples that were banked miraculously for years and years prior to the diagnosis of MDS and AML. So I think that work has to be done for us to understand the natural history. So I don't think that we know yet. But again, we'll hear from the other speakers on that.
Hi, over here.
Go ahead, I can't see who's talking.
On the other side here. Really enjoyed the talk. I was curious about if you could speculate on the thoughts of how altered splicing might drive leukemogenesis, whether there's definable and recurrent splicing targets. And then with respect to this morning's talks, I was wondering if maybe the generation of alternate splice isoforms might make these tumors more prone to immune therapies.
Yeah, it's a great, so those are all great ideas. I think, again, it's still early days. We don't have the answers. So first of all, I'd say I'm trying to keep an open mind, even though we have this amazing convergence of factors, mutually exclusive, all in this particular subcomponent of the spliceosome. Gosh, that has to be telling us that it's in fact alternative splicing that's contributing to the disease. But we need to keep an open mind to the other possibilities. There are connections between DNA repair and transcriptional regulation, splicing, and maybe these create a signal that's read out in different ways. It may connect to other biology, but again, the working hypothesis, we know for sure that they do, the mutations change splicing. I think that's unequivocal. I showed you some of our data. There's plenty of data emerging. Again, that could be true and unrelated. The general idea is if it is all about splicing, that it can be thought of like an epigenetic modification that rewires the cell. Probably not a single smoking gun, in my view. I think it's unlikely that all of these are converging to alternative splicing of the key oncogene that just seems implausible. But likely just changes the entire transcriptional state of the cell from one state to another, since these diseases the hallmark is arrested or impaired differentiation, perhaps changing it from a cell that's normally differentiating, not self-renewing, to one that's more pluripotent, self-renewing, and less prone to differentiation. But we really don't know. Those are some thoughts. And I also have no answer to your question about whether or not they're generating new antigens, but that's an interesting idea.
As I understand it, the limited number of mutations that compared to melanoma and other type of tumors.
That's right.
So, do you know if the immunogenic peptides which might be generated bind to a restricted number of HLA class I antigens?
I do not know, but I know that there's work ongoing that's similar to what you heard from Dr. Rosenberg this morning on AML, exploring the exact same hypothesis that perhaps some of these, some of the mutations that are not necessarily driver mutations, but passengers may still generate epitopes that could be used as target for immunotherapy. But there's, to my knowledge, nothing published on the peptides that are predicted, and whether or not any of them bind or generate T-cell responses. Nothing that I'm aware of. All right, I think we should go ahead, Bruce.
Just one other question is, we sort of got sold on the idea that sequencing tumors, the DNA sequencing, is the ultimate way of defining the subtype of the tumor. But it seems to me if splicing now mutations in splicing could generate a whole series of expression changes which would not be reflected in just DNA sequencing.
Well, that's...
So
yeah.
Should we think differently when we identify a sequence abnormality involving one of the splicing genes? Then we have to think that, well, we really don't understand what's downstream of this.
Yeah, I think that's fair. And many of the studies have, for this reason and others, combined multiple platforms, genomes, exomes, transcriptomes, because they give you complementary types of information. If in a disease like MDS, these mutations are so frequent, I think identifying them by looking at the genome makes sense. You're not going to miss cases. In cancers, other hemolignancies and other cancers where the frequency of these mutations maybe is not as high, there may be other ways to generate the same signature. Again, if it is mutations and splicing factors changing the transcriptome, there may be patients who have that, phenocopy of that but don't have mutations in the splicing. So I think that's a good point.